I will repeat the transcription, and we said before نحن that transcription is the copying of DNA into messenger RNA. فإذا the word transcription means to copy. Okay, what I am copying here, I am copying DNA into messenger RNA in order to synthesize the proteins. طيب, now we want to recognize or identify mRNA. What is mRNA? فإذا it is a nucleic acid. It is a nucleic acid like DNA, okay, but has some differences. And we made a table of differences between DNA and RNA. We said that RNA is single-stranded, contain U instead of T, not stable, okay, and contain sugar ribose instead of deoxyribose, and many other things. Type. Now, so what is the job or what is the function of the messenger RNA? It is to copy the DNA. And we located by methyl green DNA inside the nucleus. It gives me a green color, and pyronine, it gives the messenger RNA pink in the cytoplasm. Okay. Now, we use also radioactivity, radioactive uracil, in order also to prove that messenger RNA is made in the nucleus, then it is transferred back to the cytoplasm in order to be used to copy or to make the protein. Today I am going to discuss how the messenger RNA is made. Okay, how the messenger RNA is made in details, okay, in order to see the details of this process. Type what the one that we called as transcription. Type. Okay, messenger RNA, a copy of the gene. So it is a copy of DNA, or a copy, okay, copy of the gene. Type. Now, in the previous lesson, we noticed that DNA replicates during interphase, thus ensuring two copies of the genetic material ready to be distributed equality into two daughter cells during mitosis. In addition to DNA replication, DNA provides the genetic information to its offspring, the messenger RNA, in a process called transcription, in order for the latter RNA to lead to the protein synthesis of proteins in the cytoplasm. Okay, now let's see how exactly we form the messenger RNA. First, we have the double-stranded DNA in the nucleus. Okay, double-stranded DNA. By transcription, we make a single-stranded messenger RNA, then it can leave to the outside. Nuclear membrane, as we said before, it is only permeable to the single-stranded molecule. Now let's look at the mechanism of transcription in details. Transcription mechanism in details. Okay, the RNA polymerase uses template strand to polymerase a new complementary nucleotide to form a new messenger RNA. Type. Okay, now as you can see here, we have steps, okay, with numbers. So the numbers, okay, will tell me what happens step by step. Okay, the numbers tell me what happened step by step. Type. Now, who wants to read for me the first step, number 1? Yalla. The first step, number 1. What do I have in the beginning?
بقراها أنا.
يلا يا إيمان, please.
First step, DNA double helix twisted.
فإذا نحن we know that the DNA is in the beginning as a double helix. It is twisted. Okay, we called it double helix because it is a two strand and it is twisted. طيب, the first step to copy the gene, يعني part of this DNA, to copy a part of this DNA, what I do next directly, يلا, who will be the step 2? Okay, who will be the step 2? آية خالد خديجة يوسف. Just a small sentence. Step 2. Khalid, it is page 79. Page 79. يلا يا Khalid, you will read it.
Yes.
Okay, thank you يا Khalid. What happens in step 2?
Step 2.
You open on the page. Okay, step 2, it is above step 1, number 2, if you see it.
Unwind the nature opened by polymerase.
Okay, unwind or open. What you are doing now يا Khalid? You are unwinding it, يعني opening it. The DNA is 2 strands, صح؟ فإذا أنا in this piece, I want to move the DNA away. The 2 strands away from each other. Why? Because I want to copy one of them. Okay? I want to copy يا Khalid one of them. طيب. Look, now you have 2 strands of DNA, صح? The one up is called what?
Coding DNA strand.
Coding DNA strand. And the one down?
Template.
Or transcribed DNA strand. طيب, أنت which one you are copying يا Khalid? Template. فإذا always remember this, please. We copy the template or transcribed. The coding is the opposite. The coding is the opposite. It is used just for support. اوكي هي بتعمل سبورت للتمبليت. كيف اذا عندكم بيبر هيك رقيقه مهمه بتغطوها بنايلون مشان نحميها. الكودينج cover the template to protect it. اوكي الكودينج cover يعني cover the template. طيب now step 3. يلا who will read step 3?
السلام عليكم.
وعليكم السلام. أهلين يا آية, كيفك؟
الحمد لله تمام.
الحمد لله. Can you read يا آية step 3?
Yes.
Please.
Then RNA polymerase uses template strand to polymerase a new complementary
Nucleotides
nucleotide to form a new mRNA strand.
to form a new mRNA strand. طيب يا آية, can I ask you something please?
Yes.
Okay. آية, in the template the first letter I see مثلا is A. نحن we know the first letter on the left, you see?
عند ال DNA.
عند الدي ان اي اللي تحت اول لتر على الشمال انا شايف A.
Yes.
Okay. Now this A what is its complementary in DNA؟ شو المقابل له بالدي ان اي؟ نحن عادة مقابل الاي شو عنا؟
U.
لا بالدي ان اي. الدي ان اي ما بقلبه يو.
T
تي
المقابل له.
عادة مقابله في تي.
لا
طيب
فكرت قصدك بالار ان اي.
ايه معلش هلا حاسألك عن هي. هلا هون المسنجر ار ان اي بقدر احط بقلبه انا تي؟
لا.
شو بحط محل التي؟
يو.
بحط محل التي يو. فاذا الار ان اي بوليميريز انزيم هيدا الجرين لارج جرين will read the nucleotides of the template one by one to make the complementary أو the opposite. بس لأنه عم يعمل messenger RNA he cannot use T, he will use U. Okay? فإذا يا آية in front of A the RNA polymerase puts what؟
T.
لا عم نعمل messenger RNA. ركزي يا آية. in front of A I put U. طيب continue with me. in front of T
بالدي ان اي.
DNA contain T I put A. هلا أنا عم بعمل messenger RNA يا آية.
اوكي.
in front of C I put G. صح. كملي معي. in front of G C. in front of A U. in front of G C. in front of A U. in front of A U. in front of T A. صح؟
Yes.
هلا صار في عندك now we have a molecule of messenger RNA. مظبوط؟
Yes.
طيب now يا آية I want you to compare this messenger RNA with the template أو transcribed and with the coding. طيب let's read number 4 please يا آية if you can.
A new mRNA produced.
فإذا now you make a new mRNA. صح؟
Yes.
طيب now let's see the details. It is
complementary with the template but identical to the coding strand.
طيب تعال نشوف يا آية هل هو complementary للتمبليت؟ هل هو it is opposite directly opposite to it؟ المسنجر RNA هل هو complementary يعني it is opposite للتمبليت؟ يعني التمبليت مثلا قلت لي عندي A قلت لي opposite to it U T opposite to it A C
Yes.
opposite G G opposite C. فإذا هو مظبوط complementary.
Yeah.
طيب now يا آية compare المسنجر RNA with the coding strand. Look at the coding.
Yes.
طلعي فيها. بالمسنجر RNA first letter is what؟
A.
A مثلا. بالcoding it is also A. G
العكس.
C مع اللي فوق. أنا عم قارنها مع اللي فوق. اوكي عم قارنها مع اللي فوق. فإذا I can see that they are the same.
Yes.
بس instead of T up I have what? I have U. correct؟
Yes.
بس بدل التي يو. يعني هي ذات الكودينج بس بدل التي يو وهي complementary to the template الأصلية. طيب آخر بعدين يا آية الجملة 4 to I please
only you replace T at the end.
صح. only you
Yes.
replace T at the end and the last sentence
It transports the genetic information to the cytoplasm in order the product, the new protein.
to produce the
To
new protein.
to
هلا صار عندك يا آية. Now you have a copy of the DNA. Now you send it to the cytoplasm to build the protein you want.
Yes.
Okay?
لا.
Thank you يا آية.
Yes.
You understand it?
Yes.
Okay, thank you.
Welcome.
طيب, others now you understand what happens. فإذا the template أو transcribed DNA strand I make the complementary of it of messenger RNA, a copy. Okay, but this copy I noticed that it is the same as the coding, but T is replaced by U. T is replaced by U. Clear, guys? Clear? Any question about it? طيب, Khadija and Yusuf, what do you say? Okay, good يا Khadija. Yalla, Ahmed is with us now. طيب, for Ahmed, yeah, Ahmed, now we're discussing how we are making now the messenger RNA from the DNA. يعني the process of transcription, we are doing it now in details. For now, we will show a video on how this process happens in details. Okay, let's follow the video, please, together and see. Now this is the DNA, the part of the DNA, and I will not copy the whole DNA. I just copy part, the gene responsible to produce a specific protein. But this part only. طيب. Okay, the first step we said that we want to untwist it, to open it, لأنه هي is in the form of double helix. So it has a beginning part called the promoter, final part called terminator. Okay, promoter, terminator. Yalla, look now. Okay, this is the messenger RNA. And the enzyme here is RNA polymerase. Type. The first step, what we did, we opened the DNA into two strands. One will be called the template, or transcribed, and the other is the coding, the one that supported. Look what it will do. It'll start reading and put the opposite. So now the red sequence, you see, T, the RNA polymerase put A, G, C, A, U. Messenger RNA. Okay, until it reaches terminator, then it stops. خلاص, it is finished. Now, this happens inside the nucleus, don't forget. Now the messenger RNA is ready to leave the nucleus into the cytoplasm. Okay, I want to forward it a little. So this is the messenger RNA now leaving the nucleus to the cytoplasm in order to be used to build the proteins, and it has a sequence of bases. Those are the copy of the template or transcribed strand. Okay. طيب. عبد الرحمن with us كمان الحمد لله. Very good. Okay, I will repeat the video. Okay, I will repeat the video. Please pay attention. Okay, فإذا now I want to make a specific protein, for example insulin. I search in the DNA for the map of insulin. Not all the DNA, I want a small piece only, the gene responsible for this insulin. Okay, now an enzyme called RNA polymerase come and bind to the beginning of the gene. لأنه the gene, every gene has a promoter beginning part, what terminator final part. In any letter you write, you have the title and you have your signature in the end. Now, a large pink structure here is the RNA polymerase, the one that will read the DNA and the one that reads DNA and copy it. Okay. Now, first it opens the DNA, untwisted. Now we open the DNA. Now we have two strands in the DNA. We have the template or transcribed, the one I will copy, and we have the coding, the one that is for support only. Type. Now the RNA polymerase will start reading the bases. If it sees A, it will put in front of it U, G, C, C, G, T, A, and so on. Now the messenger RNA is built. Until I reach the end. Now I will stop, and the messenger RNA now it is ready to be used in the cytoplasm to build the protein. Okay. We get it now? MashaAllah. Hadi is also with us. Type. Any question about the process? Okay, any question about the process? Good. Type. Okay, now let's do the application number 1. Application number 1. Okay, let's focus together on this. Let's focus together on this. Type. Consider the following DNA coding strand. Please, if you have the book, put 2 lines under the word coding. Coding. Okay, let's put lines under the word coding. Type. Okay. Who wants to read? Who wants to read? يلا أحمد. هادي، يوسف، خديجة، عبد الرحمن. يلا who wants to read? طيب let me go back to Khalid. Can you read يا Khalid, or I ask Iman or Aya? طيب Khalid has a problem maybe in the internet. Iman, can you please read?
Yes.
Please.
Write the messenger
لا لا
RNA
لا from the
produced.
beginning please. لأنه the beginning is very important. Again.
Consider the following DNA coding strand.
طيب Iman, when I
NQG
say coding,
Yes.
I mean this is the one that is copied ولا the one that is similar to messenger RNA?
Copied.
لحظة, look up in the figure up.
Yes.
Let me go up a little. When I say coding, is it the one copied ولا the one that is similar to messenger RNA? Look up.
Similar to
the one that
messenger
is
RNA.
similar.
Yes.
The one that is similar. طيب, now question A will tell me write what?
Write the messenger RNA produced by the given DNA strand.
Now I want to make the messenger RNA يا إيمان. How I make it here?
ما فهمت.
Now I'm giving you the coding. صح?
Yes.
I want to make what now?
A copy?
بدي make المسنجر RNA.
Yes.
طيب نحن we take it now as a rule انه الcoding is the same as messenger RNA, but we replace T by what?
U.
By U. فإذا give me now the same strand but replace T by U, what it will be?
AGGCCUGUA.
Exactly. That's it. So this is the messenger RNA.
Yes.
فإذا whenever you have the coding, the messenger RNA is the same. Okay? Whenever I have the coding, the messenger RNA will be the same. Amma if he give me the transcribed, it will be the opposite. Okay? It will be the opposite. طيب. Who can, يلا part B. Who can give me two methods to locate DNA and RNA in the cell? DNA and RNA in the cell. يلا, I want two methods for this. How I can locate the DNA and RNA. I will say, yes يا إيمان, you were saying?
كنت عم أقول إنه فينا مثلا to locate DNA إنه add methyl green.
add methyl green.
Yes.
صح. 2.
Or for messenger RNA threonine.
To locate
Threonine.
DNA, and pyronine to locate RNA. طب الميثيل green will give me what color with DNA?
Green?
Green color. While pyronine will give me color?
Pink.
Pink. Very good. To locate RNA. Another method is to use radioactive T to locate DNA and radioactive U to locate RNA. So one of the way to identify the location is by using the radioactive. But T is found only in DNA. That's why I can use radioactive T. And radioactive U to locate the messenger or the RNA in general. To locate RNA in general. Okay, very good. طيب. Now, name the mechanism that produce messenger RNA from DNA and indicate its cellular location. Khalid, you can answer me on this. What is the name, ya Khalid, of the mechanism that produce messenger RNA from DNA? Where is Khalid?
تسفيني أقول أنا
طيب يلا name it يا إيمان.
transcription
Transcription. Where it happens يا إيمان?
in the nucleus.
In the nucleus. In the nucleus. Okay. فإذا it happens in the nucleus. طيب. Transcription, it occurs in the nucleus. طيب, part D, indicate the reason behind the need of this mechanism. Why I do this mechanism, Anna? For what? In order to do, I do this process. For what reason? I do this process in order to synthesize the proteins in the cytoplasm. In order to synthesize the proteins in the cytoplasm. So without transcription, I cannot build any proteins in the cytoplasm. I don't have its map. So I need it in order to have a map so I can work on it in the cytoplasm. طيب. Now they tell me compare in the table between this mechanism and DNA replication in a table form. When they tell me compare, I look for the similarities first, and then I look for the what? For the differences. طيب. Now, what is the similarities? Okay, let's start with the similarities. Both processes happen in the nucleus. So these are the similarities. That both happens in the nucleus. طيب, in the differences with the DNA replication, okay, I have two differences here. DNA replication uses DNA polymerase enzyme to produce DNA molecule, while transcription uses RNA polymerase to produce messenger RNA. فإذا it will be the opposite. This one will produce DNA, the other produce messenger RNA. Okay. فإذا similarities both happens in the nucleus. It differences DNA replication use DNA polymerase to produce DNA molecules, while transcription use RNA polymerase to produce messenger RNA. Any question? طيب, please Okay, I will give you a homework. It is a very important homework, and I want everyone to do it. It is page 98, number 16. Okay, page 98, number 16. I will write it down. Page 98, number 16. Please, everyone should do it, okay? And send me the answers. It's very important. Thank you for your attendance today.